Hi everyone! For Chapter 8, we will approach it a little bit differently. We will discuss the Biotechnology chapter in light of the pandemic. It will make discussing the procedures in the chapter more real if we can talk about how they're applied. First off, let's define biotech. That's the use of microorganisms to make practical products. At least this is the simplified version of a definition, but there are a lot of tools of biotechnology that are being used for diagnostics and drug development, as well as for developing vaccines. We will focus on a few tools of biotechnology and weave the process of diagnostics and vaccine development into the story that may help these techniques be more relevant to you. We will take a look at the techniques that were needed to develop the diagnostic test for COVID first, and then we'll look at the techniques to develop a vaccine after. First off, let's go through a figure that we made that describes the process of detecting the agent of COVID-19. The agent is a virus that's named SARS-CoV-2, which stands for Severe Acute Respiratory Syndrome Coronavirus number 2. This is related to SARS-1 virus that caused another serious and scary outbreak a number of years ago. This new coronavirus is a related virus. You've probably heard a lot about the diagnostic testing of COVID, perhaps even had one performed on you. This figure is illustrating a simplified version of that test. Initially, you have to obtain a sample. The most common sample is a nasal swab. Typically, to obtain a sample, the swab is inserted deep into the nostrils and rubbed to pick up any virus that may be present. Then, the swab is placed into transport media specific to the virus to transport to a testing lab where they'll be able to do the types of analysis that are needed. In the lab, RNA is isolated, which is the genetic material for this type of virus. The coronaviruses do not have a DNA genetic material, they have RNA. RNA is a little tricky to work with. We will not spend much time talking about that, but you have to handle that specimen in a specific manner or the RNA will break down. RNA is pretty finicky in the laboratory. What's important is this next step down here on the bottom, where we have to convert RNA to DNA to do the manipulations and analysis. Luckily, scientists involved in genetic engineering have known about an enzyme that will copy a piece of RNA into a complementary DNA. This enzyme was isolated from viruses such as HIV. They do what is termed reverse transcribe, meaning the enzyme will use the messenger RNA template or an RNA template and make a complementary copy as DNA. That enzyme is called reverse transcriptase. That enzyme has been exploited for genetic engineering for a lot of different reasons. It's needed for this test. The lab technicians take the viral genetic material, they convert it to DNA using reverse transcriptase, and then we're going to do a specialized version of a test called polymerase chain reaction, or PCR. In this specialized version of PCR, you're able to detect the samples as they're being amplified, as shown here. The test is called qPCR for quantitative PCR. However, sometimes they call it RT-RT-PCR for a reverse transcriptase and real-time PCR. We're going to discuss that specialized process in a bit, but first I want to spend time on the more classic polymerase chain reaction. I want to point out that there is a nice animation in Mastering Micro on PCR. This is the figure from your book. I have also linked for you in the chapter some other animations on PCR that might be helpful and a video on how it can be used in other instances. In a simple overview, what we're going to do is take a single copy of a segment of DNA and make many, many copies of it over the course of the reaction. If you look at the top up here on the left, you see that we have a gene of interest. During the PCR reactions, we're going to make copies of that gene of interest. This polymerase chain reaction, or PCR, goes through many cycles of a set of temperatures, and we'll be using specific reagents in order to run the reaction. The reactions will include your DNA, the nucleotides referred to by a scientific designation, DNTPs, which stands for deoxynucleotide triphosphates. Also, a pair of short strands of DNA called primers, which will be complementary to each end of the gene, a special heat-resistant polymerase, and buffer. First, the DNA is heated to about 94 degrees Celsius, to what's called the melting temperature. It's pretty near boiling, so that the DNA strands will separate from each other. The hydrogen bonds between the bases are broken in the heat. That's called denaturing, and that's the first step. The second step is called priming, or sometimes referred to as annealing. During priming or annealing, the little short segments of single-stranded DNA, the primers, that you've included in your reaction will stick to the complementary sequences in your target DNA sequence. After the primers stick to the complementary regions in your template DNA, the rest of the complementary strand is made. This process is termed extending. It's because the primer sequence is added to and the polymerase extends the strand. Now would be a good point to recall a few important points about DNA replication. Number one is, remember when we were talking about DNA replication, cellular enzymes would prime the reaction and lay down an RNA primer so that your polymerase could copy a complementary strand? That's the purpose of including these small segments of DNA, these primers. DNA polymerase needs to add to the 3' end of a segment of nucleic acid so that it can specifically create a complement of a template. The primers sit on complementary region that are in your gene of interest, and then the polymerase is able to use that primer and extend from it. Notice that the DNA is being made right here. An important point is that polymerases are usually heat sensitive. Normally, if we were to start the next cycle and heat to 94 degrees again, we would have denatured the polymerase along with the double-stranded DNA. But the polymerases that are used in polymerase chain reaction are isolated from thermophiles, so that the polymerases are stable when you heat them up. They do not denature. Actually, the extension step is performed at a pretty high temperature. It's about 72 degrees Celsius. That's way too hot for our own polymerases and the polymerases of other mesophilic organisms, but this is a good temperature for the polymerases from a thermophile. In PCR, these 3 temperature steps are repeated over and over again. Denature, primer anneal, extend. Denature, anneal, extend. Let's review once more. What did we include in our reaction tubes? Well, of course, we have to have our gene of interest. We also need all of the DNTPs, the nucleotides, the G, A, T, and C. We have to have our primer set. Also, the special DNA polymerase like TAC, TAQ, that is heat stable. Finally, you need the correct buffer. Once everything is mixed in the correct amounts and the tube is placed in a thermocycler, which is the machine that can be programmed to go through these temperatures in many cycles, usually around 30 times. The first cycle, you could start from 1 copy of your gene of interest, and when the cycle is complete, there would be 2 copies of the initial template. Each cycle will double the DNA template. In the end of 30 cycles, doubling the DNA in each cycle will permit the production or amplification of your one template to millions and millions of cycles, millions of copies. That's PCR in a nutshell. It has become an extremely useful, a key step in many other procedures. It's used in diagnostics, genetic engineering, research, and more. I recommend watching the animations, which will help you a lot in understanding how this works. It's a very powerful tool. The person who figured this process out, Dr. Kary Mullis, lived in San Diego. He won the Nobel Prize for his discovery, which is pretty amazing since techniques are not often the topic of a Nobel Prize. But this technique has been instrumental in moving very many other areas of science forward due to its extreme usefulness. So, after you've made all of these fragments of DNA, you need to do something to see them. You can't look at a PCR tube and see that there are more DNA molecules in it than you started with. In this classic method of PCR, once you have all of those DNA fragments, you would have to analyze them. Typically, that's accomplished using what's called an agarose gel in a process called gel electrophoresis. Essentially, what you can see in this little figure here is that we have this gel made out of an agarose, which is basically a purified form of the agar that we use for media in the microlab. This very pure type of agarose is melted and poured into a mold. There will be some wells that have been formed as the agarose gel solidifies. The wells are where you load the sample. There are these little rectangular pits in the gel where you add sample. Then you subject the gel to an electric field which causes the DNA to migrate or move towards the positive electrode. This is because DNA has an overall negative charge. Think of all those phosphates in the molecule. The DNA pieces separate by size. The smallest ones get to the bottom much faster than the larger ones that have a little more trouble migrating through the gel matrix. Once the DNA moves through the gel for some time, you can analyze the bands and the band pattern to get information. You can certainly figure out what you need to from gel electrophoresis of PCR reaction, but it takes time, effort, and it has the potential for several things to go wrong due to human error. Of course, it's less expensive than the specialized version of PCR I am about to describe that's used for the COVID antigen test. The real-time PCR, sometimes called quantitative PCR, is a PCR that can be read as the samples are being copied, hence the real-time designation of this version of PCR. It requires a special primer. For RT-PCR, the regular primer set must include as well, but there's also a special primer with a fluorescent molecule and a quencher molecule. If the pair, the fluorescent and the quencher molecules, are near each other, there is no fluorescence. If they're separated, the fluorescent molecule gives off light. In this example, the special primer is called the TACMAN probe. TACMAN is a brand that's commonly used for these reactions. The idea is that you have a fluorescent molecule and the quencher that tamps down the fluorescence. So the primer won't fluoresce when the quencher is next to the fluorescent molecule. However, during amplification, during the copying of the DNA polymerase will come to the area where the primer is sitting with the fluorescent molecule, and the polymerase knocks off the fluorescent molecule and allows it to emit light. Every time you amplify a piece of DNA, more of this fluorescent molecule is being released. Your sample tube gets brighter and brighter, and the emitted light can be detected if you have the right PCR machine. The benefit of this particular type of PCR is that it's really fast. You don't have to run a gel step, so that end result is obtained much faster. There's fewer steps, so you have less room for human error. Also, the reaction itself is more sensitive than conventional PCR because the light detectors are very sensitive to the fluorescent signal. Of course, you need a special fluorescent primer, and you need this special light detecting thermocycler, which is far more expensive than a normal thermocycler. So that's all part of the package. The last thing that I want to mention to you is that we have to make a cDNA, which is the DNA copy of an RNA molecule. It's accomplished with the viral enzyme called reverse transcriptase that has been used by genetic engineers for some time. When we're trying to detect an RNA virus, such as SARS-CoV-2, the agent of COVID-19, we have to reverse transcribe first in order to do the real-time PCR, which is amplification of DNA. We need to make a DNA template from the RNA genome. This reverse transcriptase step gets combined with that real-time PCR or qPCR. You will see this type of combo test being referred to as RT-PCR for reverse transcriptase, real-time PCR, or sometimes RT-qPCR for reverse transcriptase quantitative PCR. For the COVID diagnostic test, the viral RNA that was obtained from the swab of the infected person is reverse transcribed in that first stage so that you have a strand of DNA. Then the reaction will replicate that strand of DNA. From that step all the way forward, the real-time or quantitative PCR is occurring. Positive samples are detected with the qPCR machine and reported out. An example report is seen in this graphic, and the signals rising up show the positive samples. And you can tell the relative amount of the initial template depending on how these lines are arranged on the graph. That's the quantitative part of the test procedure. The samples that don't have RNA or a template never start fluorescing. You have a bunch of negative signal, which you can see on the bottom of the graphic. In a nutshell, what I want you to remember for this first part of Chapter 8 material is the concept that RNA can be converted to DNA using a tool of genetic engineering, the reverse transcriptase enzyme. The DNA made from an RNA template is called cDNA for complementary DNA. And understand how the PCR works and what gel electrophoresis does. And finally, how RT-qPCR works. The next video for Chapter 8 will concern DNA sequencing and cloning genes to make proteins, aka genetic engineering. Okay, that's all for now. See you next time.